All right, we'll give people another minute to come back. All right, welcome back, everyone. So we started the session, or the evening, I should say, looking at organ development, and we jumped right into stem cell and gene editing therapies. But we're gonna finish the evening with a focus on medical therapies, and it's really an area that we're quite interested in because there are a lot of advances in medical therapies in neonatology and pediatric disorders, and we'd like to start thinking about translating that approach into the fetus. And we're really lucky and honored and thankful that Professor John Day has agreed to start this session with us. And Professor Day is a neurologist and an expert in neuromuscular disorders, and he runs the neuromuscular disorder program here at LPCH in Stanford. And he's really a leading researcher in disorders such as ALS and muscular dystrophy. And there's a focus in prenatal medicine on SMA and many other disorders, but we're really thankful that Professor Day has agreed to come and share his thoughts about sort of medical therapies for neuromuscular disorders and maybe others. So without further ado, I'll let you take it from there. Thank you.
Great, thanks, Yair, and thanks for inviting me to this really exciting symposium. It's really amazing to hear everything that's going on. So let me attempt to share my screen. And so it's great to be able to talk about our experience in working on spinal muscular atrophy as a prototype, we always believed, for treating neurogenetic and neurodevelopmental neurodegenerative disorders in neonates. Nobody goes into neurology to go into neuromuscular medicine. Everybody wants to study the brain, but the advantage of studying neuromuscular medicine is that we can actually biopsy the tissue. It's allowed us to be much more specific, and it's led to several different discoveries over the last several decades of identifying genetic causes. So we literally just, you're not supposed to be able to read this, but just in terms of muscular dystrophies, we literally now have hundreds of identified genetic causes, which is unusual in neurology. Neurology, for the most part, is still clinical diagnostic realm. And so being able to identify these disorders gives us a target for therapy. These disorders are neuromuscular, but that doesn't mean they're limited to affecting nerve and muscle. And so we're going on to investigate these disorders because we can determine the effects of treatment on the nerve and muscle aspect of the disease, but also be able to then use that method of treatment to try to reverse and treat various central nervous system disorders as well. So a lot of that work is ongoing, but we started with spinal muscular atrophy for very clear reasons. And I give credit to Kurt Fischbeck and Jerry Fischbach at the NIH around the turn of the century, who pointed out that SMA was indeed an excellent prototype for developing novel treatments. And that was for several reasons. I mean, it was a fairly straightforward genetic recessive inheritance. So it seemed like it would be quite easily treated by gene replacement. It was obviously genetically well-defined. I'll talk a little bit more about that, and genetically homogeneous. The most severe form is the most common form. So it's got a very clear phenotype. These children typically, classically look normal at birth, develop symptoms in the first couple of months of life, and about 50% of them die within the first 10 months, with the remainder dying typically within the first 2 years. So it's quite prevalent as far as genetic neurological disorders go, with a carrier frequency of about 1 in 40 and a frequency of at birth of about 1 in 10,000. It didn't look like it was toxic to overexpress the gene. That's going to be revised as we've learned somewhat more recently, that there probably are some concerns about overexpression. But because of this rapid time course, it obviously allowed us to see the effects. It's a very restricted phenotype. So as opposed to many other forms of muscular dystrophy and neuromuscular disease, it didn't really have any severe cardiovascular, GI, or other metabolic components. And importantly, it did not affect the rest of the central nervous system. So these kids were quite alert, though they were obviously devastated by their neuromuscular loss. So it was really pointedly directed at developing this as a prototype, and it's been a couple of decades now of work on this that has allowed this to move forward in the therapeutic realm. So what is spinal muscular atrophy? For those who don't remember your neuroanatomy, it involves the anterior horn cell, or these cells in the spinal cord that are the neurons that go out to muscle and control muscle. So at least for first estimates, these cells are the primary cell affected. So although the causative gene is ubiquitously expressed throughout the body, it is really a disease limited to these anterior horn cells in the spinal cord. And those cells are quite important. I mean, even though there's a relatively small population of them, because they go to many muscle fibers, so that each neuron goes to many fibers. Here in this cartoon, it's shown going to 5 fibers. That's far and away from the truth. Here's a single motor neuron in the spinal cord, and it's going to, in this case, several hundred muscle fibers in the soleus of this rat. In humans, one motor neuron will go to several thousand muscle fibers. So obviously, these are important cells, and consequently, an inadequate number of them leads to devastating effects. The genetics, as I pointed out, are quite straightforward. It's a recessive disease, so that they eventually, in the 1990s, identified the gene responsible for this disorder, SMN1, or survival motor neuron 1. And it's normally expressed at quite high levels, as I pointed out, ubiquitously, but in SMA, it's knocked out, so it's a simple recessive disorder. What makes it an even better target, though, is there's the unusual circumstance where there's a second gene that makes the identical protein. There's nothing different in terms of the protein produced by these two genes, SMN1 and SMN2. This is due to an inverted duplication on chromosome 5. But this second gene has a single nucleotide polymorphism at the 5' end of exon 7 that alters the splicing of exon 7, so that the majority of transcripts from SMN2 are devoid of exon 7, and that leads to no effective protein production. There is a small amount of transcript that does include exon 7. Here it says 20%. It's probably closer to 5 to 10% in more recent assays. But nonetheless, it does produce a little. If there's complete absence of SMN protein, it's an embryonically lethal condition. But if there's a little bit of protein generated from SMN2, then the individual can survive. And so this is obviously in a non-SMA patient. You're getting expression from both the SMN1 and SMN2 genes, but you can see that the vast majority of the SMN2 gene is missing exon 7 at the RNA level, and consequently is going to lead to very little protein production. And in an SMA individual, there's no SMN1 expressed, but there's still SMN2 expressed. And that small amount of protein production is enough to get the child to be able to survive. It's further complicated genetically because there's SMN1, as I mentioned, on chromosome 5, and that's missing, and that causes SMA. But the number of copies of SMN2 is quite variable. So on one allele, there can be as many as 3 or 4 copies. So any one individual can carry as many as from 0 to 6 copies of SMN2. As long as there's a single copy of SMN1, it doesn't matter how many copies of SMN2 you have. But if you have multiple copies of SMN2, that obviously modifies the phenotype to some extent. So in a carrier with a single copy of SMN1, there's no phenotype observable whatsoever. If you have, as I mentioned, 0 copies of SMN1 or SMN2, it's embryonically lethal. But if you have at least 1 copy of SMN2, these children are generally born, but they're severely affected at birth. So they're born with contractures and severe weakness of ventilation and other muscles even at birth, and live for only a matter of months. But if you have 2 copies of SMN2, which is the most common genetic situation, these are the children I was describing with what's termed classic SMA type 1, which fortunately we don't see anymore now. And then the more copies of SMN2 you have, the milder the phenotype. This just confirms that. This is a haplotype, a histogram showing the occurrence of SMN2 copy numbers and demonstrating that the phenotype does quite closely model the number of copies or follow the number of copies of SMN2, although there are other genetic modifiers that have been identified as well. Okay, so the weakness is very characteristic. There's a very specific pattern. These kids can be diagnosed very easily at birth. This is a picture of the type 1 infant, so-called Verdnig-Hoffmann disease in the pre-genetic era, it was known as. And I mentioned these children typically die about a year of age. The milder form in patients with typically 3 copies of SMN2 or type 2 SMA is noticeably milder. So these children live into adulthood, although they generally succumb without some form of treatment in the early adult years, and typically have onset of weakness evident in the latter part of the first year or 2 of life. And then it gets even milder with this so-called Kuckelberg-Wieslander form of or type 3 SMA, where they have an onset of symptoms typically after 18 months of age. These patients will have a normal lifespan, but they typically do lose the ability to walk at some point. So further investigation of the genetics of this in the late 1990s and early 2000s demonstrated that there was an intronic splicing silencer in exon 7 that could be manipulated to force inclusion of exon 7. Remember, it's exon 7 that is alternatively spliced in SMN2. So in all SMA patients that are missing SMN1, they all have a copy of SMN2. And by altering the splicing of exon 7, it's possible then to force inclusion of that exon and then generate more protein. The first attempt at that was a development of an antisense oligonucleotide. This was done by Adrian Craner at Cold Spring Harbor, working with Frank Bennett from what was then known as ISIS Pharmaceuticals. For some reason, they changed their name to Iona subsequently. But this antisense was developed and shown in cell models to effectively increase the splicing inclusion of exon 7. And we dosed the first infant with this treatment in 2013. That obviously led to development of this as a now commercially available treatment. But that isn't the only approach that was taken. There was the work of Brian Casper and Jerry Mendel at Nationwide developed an AAV approach for SMN gene replacement treatment. And they showed in the early 2015 or so that this seemed to work. And then we were able to dose the first patient with the commercially available form of this in the phase 3 trial in 2017. And then there was a third approach developed using a small molecule that came out of a high throughput screen assay from PTC Therapeutics that also was able to effectively alter splicing. So all three of these treatments now work. And just to show you the benefits, so I just chose one video of one of the patients. So this ends up being the patient that got the AAV treatment. Just to show you that it's not that we were treating before onset of weakness. So this is an almost 3-month-old girl who had a diagnosis actually made in utero, but came to us for treatment, as I said, in 2017, and she's obviously quite clearly weak here before treatment. So this is before treatment. All three of the treatments I'm going to show you, that the infants look very similar at the time of treatment. So they were definitely affected at the time that the treatment was initiated. And here's the child that was treated with antisense oligonucleotide several years later. I actually just saw her back in clinic yesterday, and she's now 9 years old and in 3rd grade and doing extremely well. Again, these kids have typically normal cognitive and behavioral and emotional function. So here's this child that you're seeing the video of above, 18 months after treatment. So children, the rule with SMA type 1 is that the children are the strongest you'll ever see them the first day you see them, so you never see them gain strength. And obviously that isn't the case here, where she's sitting on her own and behaving quite functionally. So there was obviously a population of neurons here that could come back and begin functioning again. This is the first infant treated in the US with the oral compound, the small molecule approach, and he is doing extremely well now as well, treated in 2017 as well. So all three of these treatments have now been approved. And this just shows you that I'm not just picking out individual cases, so that this is our paper on the AAV phase 3 trial, showing that basically every patient improves and improves quite dramatically after gene restoration. Not everyone, there are some patients here down at the bottom who did not do very well. So there's still some variability. This shows you the results in the nusinersen antisense oligonucleotide treatment, making the important point that it's always better to treat younger kids that are less affected, so that the brown and blue lines here show the responses of children who are treated in the first month of life, as opposed to these other lines are patients that were treated between ages 2 and 6 months of life. So there really is a narrow window here to start effective treatment. And we're scrambling to try to get these kids now dosed as quickly as we can. But obviously we have some effective treatments. Just to make the point that these treatments still have some unknown problems and some known problems. So the problem with antisense oligonucleotide is the current versions of them do not cross the blood-brain barrier, so they have to be administered intrathecally. There's variable biodistribution. There are problems of some neurons not being adequately treated. There are issues about the dosing, and then there's a lack of systemic administration. While the primary phenotype is motor neuron disease, there are other phenotypes involved, and so we're continuing to explore additional treatments. The immunologic side of AAV administration, we're using 1.1 × 10⁻14 viral genomes per kilogram. So it's a massive dose of virus in these little infants. And that has its own concerns with regard to immunologic effects, and obviously the concern about biodistribution again, about not fully treating all cells, and then the inability to redose because of the immune reaction. And there are also questions about risdiplam. So all of these issues are constraining us, but we're moving forward. Newborn screening was initiated 18 months ago in California, and in that time, we've had 6 referrals to Stanford. There have been close to 30 patients identified in California in that time. For out of our 6, 2 were diagnosed actually in utero. But the point I wanted to leave you with was that both of them still show signs of weakness after dosing. They both had symptoms before they could get dosed. And of the 5 patients who had 2 copies of SMN2, all of them had signs of weakness on first exam as well, and continue to show evidence of weakness. So there's still an unmet need, which moves us into the concept of trying to move this treatment into in utero gene correction treatments. And obviously there's a lot of preclinical work that has to be done there. So in conclusion, all 3 of these treatments work. We think that Fischbeck and Fischbach were vindicated in their selection of SMA as a prototype, but now we're moving on to treat other neurogenetic diseases. But there is an unmet need, and we think that in utero treatment really would be essential for patients, at least if we can find a safe way to do that. So thanks again for inviting me. It was great to be here.
Thank you.
Oh,
Thank
I did want to point out the team that did this, though. I have to give the credit where credit's due, and especially mention Carolina Tessi-Rocha, who's the director of the Neuromuscular Clinic at Packard, and the rest of our team. So thank you very much.
Thank you so much, John. That was outstanding, and definitely gets folks thinking. Our last but certainly not least speaker, and you know, as we were thinking about the symposium and planning for it, we wanted to sort of start thinking outside the box, and we invited Professor Paul Wender, who's the Bergstrom Professor of Chemistry, and who's an expert in where chemistry meets systems biology, and thinking about how, with all of these disorders, as we're starting to think about the future, one of the challenges, of course, is getting various therapies across different barriers, whether it's, you know, through the mother, whether it's through the placenta, whether it's through different organs in the fetus, and how do we get these therapeutic modalities into the areas or within the fetus that we're really targeting. And Professor Wender has spent a long time thinking about these kinds of problems, and maybe he can share with us a few of the historical solutions that he's come up with just to get us thinking about things the right way. So Professor Wender, thank you so much for your time, and we look forward to your presentation.
So hopefully this is all up and visible. Great, thank you. So thank you, Yair. And I'd like to start by actually congratulating you and Yasser and those involved with the Dunlevie Center. This is really exciting. And it's so exciting to see you reaching out to make connections. You know, the spirit of this meeting today, I think, is very important. Because many of the problems that we have, as everyone knows, are not monodisciplinary problems. They're translational, they're multidisciplinary. And that's an approach that I think has not been emphasized enough, albeit Stanford is, you know, one of the best places for this. I came here from Harvard many years ago, and I came here simply because of this translational opportunity. And it's not only on campus, it's external. which makes this a very exciting place to be. This first slide is here just simply to acknowledge the people who make great science happen. And also it kind of subliminally illustrates the nature of our research. We find problems, clinical problems, and we kind of extract from them a chemical understanding and a biological understanding of how we might be able to help out. And then we try to grow that into a platform technology. So at the end of the day, we serve up technologies that could enable, enhance many of the things that are going on campus. And this was one of the original reasons why I moved here from Harvard. As a result of that, you could see that we're involved with many academic and industrial collaborations. There are over 30 at this point in time and counting. Some people who are on this Zoom meeting, like Vittorio Sebastiano and others, their names are seen here, and a lot externally. And what we do sometimes with our technology is carry it as far as we can on campus through collaborations and also through external collaborations with many, many laboratories, Jerry Zak at UCLA, Tony Fauci's laboratory at NIAID, and others, and develop those collaborations, then looking at disease-specific applications. So this often leads to the creation of companies or licensing or options to external entities that have deeper pockets than we have to pursue the science. So I'm going to tell you a little bit, as Yar has indicated, a little bit about some of the things that would be one of these platform technologies that we work on. And this is delivery systems. And a good way of thinking about this is that pretty much anything we do with respect to therapies or diagnostics, and even to a certain extent prevention, and certainly research, involves getting things to, into, and across biological barriers. We have been conditioned historically to think about this in a very restrictive way. And if you look at the upper right-hand corner of this slide, you see a chart which indicates how we've been designing drugs for the last many, many years, trying to keep to a log P that's roughly 2 to 4, because we know that that would give us solubility and also permeability in these systems. As a consequence of that, we have left out a whole lot of what is possible. Other molecules, big molecules, small molecules, molecules that are not water soluble, molecules that are too water soluble. We've left them out of the consideration. And a way of thinking about how could we bring all of this chemical space into play in terms of therapy and diagnostics is to think about delivery systems. So that's what I'm going to tell you a little bit about today. And the first of these stories that you see here, and I'm going to see if I could remove. Okay, the first of these is a story that started in 1997, inspired by work by Frankel and Pablo Green and Lowenstein on HIV TAT protein. They observed, they were not interested in delivery, but they observed that HIV TAT, unlike most proteins, actually gets into cells. And more importantly, or equally importantly, is it goes to the nucleus. So it has a nuclear localization signal on it. Now, what we found interesting about this, and a lot of other people found interesting, is that presumably the basis for this was this sequence of ARG, lys, lys, ARG, ARG, glue, ARG, ARG, ARG. 8 of those 9 amino acids are positively charged. How could something that is so polar cross a nonpolar membrane? So that was the question that was posed to us by this research that we took up in 1997. We basically reverse engineered this so-called TAT9 bird, did truncations, alanine scans, modifications of various residues, and came to the conclusion after about 2 to 3 years of research, that it's the number and spatial array of guanidinium groups that enable uptake. And this is absolutely fascinating because here we have an entity, a peptide, an octopeptide, that has the solubility of table salt in water, but yet it gets across biological barriers. So we knew what the secret sauce was, the special structural features of this that enabled passage across barriers, but we didn't know the mechanism for that. Okay, so what's important is the number and spatial array of guanidinium groups. And let's now think about how that might lead to, how that might lead to uptake. So here's an experiment. It's a very simplistic experiment, a classical experiment, but I think this is the seeing is believing part. If we attach fluorescein to an octaornithine, so we have 8 positively charged amine groups in that system, and we partition it between 2 solutions, one water and one octanol representing the dielectric of a membrane, it stays in the water layer. And that's what you would expect. It's a polar molecule. If we do the same thing with octaarginine, it too stays in the water layer. If we mix a cell membrane lipid, a fatty acid, or even a sulfate or a phosphate in with that, in the case of the octaornithine, it stays in the water layer. In the case of the octaarginine, it actually goes into the layer, the octanol layer that has the dielectric of a membrane. This is fascinating. It's a molecule that changes its physical properties when presented with membrane lipids. So what we think is going on here is that while there is a positive charge on the ornithine, H positive charges on the octaornithine, that electrostatically would associate with a negatively charged lipid, there's only one hydrogen bond that could form between those 2 entities. Whereas with the guanidinium group, you could get 2 hydrogen bonds in the electrostatic association. And the reason why 8 is important, or numbers in between, this is a Goldilocks kind of thing, too few, too many doesn't work, but in between is just fine, is because you need a certain number to stick to these charges on the surface of the cell, and also a certain number so you could retreat from that stickiness once the molecule gets inside the cell. So this octaarginine, therefore, basically self-lipidates through electrostatic associations with lipids, moves in, and then disassociates on the inside. And it's driven inward by the membrane polarization. You could hyperpolarize a cell, things move in faster. You could reduce the polarity of the cell membrane, and they move in more slowly. It's an absolutely fascinating system that nature has developed, and we just had the benefit of observing what nature had been doing and converting it into a drug delivery system. Okay, so with that, once you know how something works, you could begin to carry a lot of things into cells. And I'm not going to dwell on this because of time concerns, but we could take small molecules, peptides, transporters, plasmid DNA, protein nucleic acids, messenger RNA, siRNA, liposomes, quantum dots, and carry these things across biological barriers. Plasmid DNA early on with Paul Wender in the medical school. So it's fairly good. And then the translation now into things that one might do came through the collaboration with Nelson Tang in the medical school. He's obviously in gynecology, and so we were looking at ways in which we could treat ovarian cancer using this technology. He was one of the first to treat women with ovarian cancer with the drug Taxol. And what's observed there is that women become resistant after a period of time to Taxol. And so the idea that we had, because that resistance is based on PGP export, is simply to attach our transporter to Taxol and let that get around the export pump that creates the resistance, let it get around that into a cell where it can be released through a cleavage of a disulfide bond, get to its microtubule target, and overcome resistance of Taxol with the Taxol conjugate. And this works extremely well. And this was the basis For example, for one of my coworkers who just 3 or 4 years ago finished her PhD, and she's now CEO of a company called N1 Life that's using this kind of technology to go after resistant cancer. At the same time, with the Cegelski group, we showed that you could attach these types of transporters to antibiotics and make them orders of magnitude more effective than the parent system, vancomycin in this particular case. So that was picked up by a group of Israeli scientists, and it's part of a new company. They hope to go into clinical trials early next year. Now, with the polyanion rage that's been taking place over the last 10 to 15 years with RNA, siRNA, and messenger RNA, and plasmid DNA, and CRISPR-Cas9, we decided that it would be good to see if we could get those things to work inside of cells. And so we took that on with collaborations with the Weymouth group, Levy, Blish, Magetti, Chang, Sebastiano groups here. Very simply put, for this, because we have a bigger cargo, we need bigger transporters. And so we developed a one-step way of making oligomeric materials that will take a polyanion, which by itself does not get into cells, but when complexed with this polycation, can be carried into the cells. It's a lipidated polycation, can be carried into cells. And once it gets inside of a cell, these nitrogens that are attached to ester linkages become amides, and therefore they go from a charge to a neutral system, releasing the polyanionic cargo that they had carried in. So we refer to these as charge-altering releasable transporters, and they work with siRNA, with mRNA, with plasmid DNA, with CRISPR-Cas9. Here's an example of that with various cell types that we're getting 90%, 95% transfection efficiencies. If we use the mRNA that codes for luciferase and inject that into muscle, we get basically luciferase expression, and we could count photons, so we could quantify the effect by treating the animals with luciferin, this substrate for luciferase. A tail vein injection, we see basically this material carries the messenger RNA to the spleen principally in these cases. If you could do a muscle injection and get one's own cells to make the protein from messenger RNA, you could obviously use this for a COVID vaccine. So with the Levy and Weymouth laboratories, while we were restricted in what we could do in the lab last year, we were allowed to do COVID type research. So we made a COVID vaccine that really works well, and it looks to be as robust as the Moderna vaccine. So that's going forward for both prophylactic as well as therapeutic applications. A discovery of, I think, considerable significance. There's a theory behind this, but yet it was surprising to see this, is that by changing the cart, the delivery system, we change where it goes after a tail vein injection. So with one cart, we get 99% spleen selective uptake, and obviously one finds a lot of immune cells there. And by changing the cart, we go 91% to the lung. And changing it yet again, we get systemic delivery, which is significant because we're obviously getting across the blood-brain barrier in those cases. So we're not using a targeting peptide or anything of that type. This is intrinsic to the cart itself that's determining where it goes. We could use this to train the immune system. Work done here with a A20 lymphoma tumor with Ron Levy, where we treat with the OVA mRNA cart and with a TLR agonist to stimulate the immune system. These carts are non-immunogenic, so we need a stimulator, and we actually get cures in animals of up to 80%, medium-sized tumors. In a 2-tumor model, we could do an intratumoral injection, and we get eradication not only of the tumor that was injected, but also the tumor that was not injected. And we know that the material that we inject stays where it's injected. So this is an example of an abscopal effect where we're training the immune system to learn to target a certain type of tumor, and then it surveils the body for that tumor and clears it. So metastatic disease. With Catherine Blish, we've been able to use CARTs to manufacture CAR NK cells. And we're doing this in collaboration with the Fauci lab and Jerry Zak and colleagues at UCLA in efforts to eradicate HIV/AIDS through a kick and kill type of strategy. So stimulating the infected cells to get them to express proteins and then using NK cells to clear. And we're at the point now where we're basically seeing treatment interruption, and obviously permanent treatment interruption would be a functional cure. All right, so that's a quick trip through some of the delivery modalities that we've been working on, molecular transporters for small molecules, CARTs for polyanion delivery. We've been able to show that we could get things across a variety of barriers, blood-brain barriers, skin, ocular, buccal barriers, transporting antibiotics, overcoming resistance. The various polyanions that you all are working with can be used. CRISPR-Cas9 can be delivered, with the Cas being an oligonucleotide. We see organ and cell selectivity, and we can use this to manufacture cells ex vivo, and we hope that the next stage will be to do this in vivo. On the far left are some of the collaborators whose work I described, and the columns in between are coworkers who are responsible for doing this work. Thank you.
Thank you so much. That was fascinating, and it's amazing, just amazing work. All right, so in the few minutes that we have left, we certainly have time for some questions. Maybe we can, you could stop sharing if you don't mind, Paul, and then we can have people ask questions. It looks like there might be not really a question, but a reminder from Giannina about to wrap up and to take a poll. But please, if anybody has any questions, we do have a minute or two.
I'll ask a question. This is Virginia Wynn to Dr. Wender. Any chance that you've looked at the delivery systems in pregnant animals to see whether any of these CARTs cross the placenta barrier?
Yeah, it's an excellent question, Virginia, and Yair and Yasser and Vittorio Sebastiano. We've been talking about those kinds of things. And for us, what makes for innovation is finding new cargoes, new barriers, new therapeutic opportunities. So the placental barrier is clearly one that we know relatively little about. We did some work with mouse pups for a while, but basically we're just dealing with the bandwidth problem right now. We have, with all these collaborations, things just haven't, we haven't had enough time to do what we need to do. But this is why I'm excited about the Dunlevie Center and reaching out for collaborations, because that kind of puts a little bit of pressure on us to see what we could do along those lines. So it's an excellent consideration. Excellent.
I promised to finish on time, which means in about 2 minutes. So I don't know if Yasser or Leslie, and I see you guys are on, if you have final comments to wrap us up here.
I would love to open the floor to Leslie, who has been so instrumental in putting the vision and mission of the Dunlevie Center together. Leslie, could you say a few words? I don't know if Leslie's still on, but I will say, just listening to our speakers today, I've been at Stanford since I was an intern, and it's amazing how siloed one gets, and how little truly one knows of the incredible wealth of intellect that we need to tap into to move this discipline forward. And so much of that resonated on each and every one of these talks. I really can't thank the speakers enough. We hope this is just the beginning. The Dunlevie Center, honestly, was just launched a few months ago. So we have a long way to go, but what a beginning it has been. And I and the whole team are just so excited and so grateful for our collaborators and their willingness to participate. I really want to thank Anna Gearson and Janina Ong for all the work they did on putting this conference, this symposium together, as well as Roger Peterson and Virginia Wynn and Vittorio Sebastiano for their brainstorming with us about how to design and kind of have an agenda that made sense and flowed well. And then obviously Yair, who has worked tirelessly ever since he was a fellow in this area of prenatal diagnostics and fetal therapy. This is your passion, Yair, and you've just led the way into a new era at Packard. When I think back at what we had just a few years ago, where we weren't doing any fetal therapy, and now with you and Carl working together, and Susan in the fetal center, the things that we are doing just were unimaginable. So thank you all. Thank you for hanging here with us, and look forward to much more in the future.
Thank you, Alisar, and thank you to your leadership and in all things MFM, and especially via this center that has brought us all together tonight. And we look forward to ongoing collaborations that hopefully will come out of this evening in the very near future. So thank you, everyone. Have a great night. We're 1 minute over the time limit, but hopefully you'll forgive us just given the quality of the event. So have a wonderful evening, and we look forward to ongoing discussions.
Bye.